Now we will begin Session 6, Deciphering the Mysterious Function and Regulation of Sleep. I'd like to introduce our session chair, Dr. YANAGISAWA Masashi from the University of Tsukuba, who will be guiding us through the session. Our speaker for this session is Dr. Sara Aton from the University of Michigan, Ann Arbor. Dr. Yanagisawa, over to you.
Okay, thank you. Changing the gear a little bit, we too will talk about sleep. And instead of general introduction of our team, in the interest of time, I chose to introduce you my own lab's story in the first 15 minutes or so. So I will talk about the mysterious regulation of sleep, and Sara will talk about one of the functions of sleep, which is memory consolidation. So our Moonshot team is interested in sleep and hibernation, depicted here in the phylogeny of the mammalian species. Where is my pointer? Oh, here it is. Okay, good. So I'm not going to talk about hibernation today, but if you look at this graph, the vertical axis is, these are each terrestrial mammalian species in the order of phylogeny. That's important. And the vertical axis represents the average time of sleep per day or per night. Two important messages from this graph. One is everybody sleeps. There is no species known not to sleep. That's already a mystery. Sleep is clearly a very risky behavior in every sense, but why it's so sternly conserved through the phylogeny. Another question is, if you know, even from the distance, you can see some correlation of sleep time with the phylogeny, which these authors name phylogenetic signal in daily sleep time, which implies that you know, this daily set point of sleep time, sleep amount, is written somewhere in their DNA. But how it's set, it's completely unknown. So our Moonshot team is basically, as its core, our own institution, which we call IIIS. So all of these PIs more or less participate in this Moonshot project together with domestic and international collaborators. So as I already said, there are two, I would say, big questions in the field. Why does everybody sleep? What's the function, such an important function of sleep which surpasses the risk? And another question is how it's regulated. So in terms of the regulation of sleep, one subfield within this question has been skyrocketing in the last 10 years or so, mainly due to optogenetics and chemogenetics type of, you know, new neurophysiological tools. So the current concept of this, you know, sleep-wake switching circuitry, which is implemented at the level of neural circuitry. So the current concept is that there are multiple wake-promoting neuronal groups or neuronal networks. On the other hand, there are sleep-promoting neural networks. And a little bit of oversimplification, but the common theme is that these two groups of circuitry compete with each other. They are, you know, either directly or indirectly inhibiting with each other, constituting a flip-flop-like switch, metastable switch. So pretty much everybody in the field agrees that this whole brain sleep-wake switching machinery is, as I said, implemented in a neural circuitry. However, if there is such a switch, what's the finger which pushes the switch? That's a bigger and a deeper question. So in the textbook level, the sleep pressure, so-called, this is a propensity to go to sleep, that's kind of encoded by the two processes. One is a circadian clock-driven process, which I'm not going to talk about except for one slide today. And the main theme is this what's called sleep homeostatic process, which dictates the following. So this is a kind of depiction of sleep homeostatic process allegory with a Japanese bamboo thing in the Japanese gardens. So this bamboo tube is upwards and the water is trickling in very slowly. That's wakefulness. And after, say 15 hours or so in humans, this is being filled up, and then suddenly it will be going downwards. And this is neural circuitry-based switching, and water will flow out and then go back to the wakefulness. So this is nicely depicting the mechanism of sleep-wake switching machinery, which is in engineering terms called the relaxation oscillator. However, what is this water? What is this bamboo tube, which means counter or accumulator? What is the transduction mechanism in which the information that water is filled up is transmitted to the switching machinery? So these are essentially completely unknown. So nearly 10 years ago, we launched this forward genetics approach of screening ENU random mutagenized mice by EEG-EMG-based real polysomnographic phenotyping. And this is the simplest possible dominant screen approach in which we could map the potential gene, causal gene in N2 backcross generation. So this is said easy, but screening tons of these mice, several thousands of these mice by EEG-EMG phenotyping, it's not an easy task. It's a really labor-intensive thing. But we continue to do that. And one of the first mutations we mapped was called sleepy. These mice are literally sleepier than normal mice. They do sleep a lot more than wild-type mice. And despite they sleep a lot, they are still sleepier. This vertical axis is an important index. So this is a delta power, so-called. So it's a low frequency component of EEG signal during the non-REM sleep, which is known to be the best marker so far for the sleep depth, as well as at that time ongoing level of sleep need or sleep pressure. So these mice are, sleep a lot, but despite sleeping a lot, they are still sleepier. This is phenotypically very very similar to the human disease called idiopathic hypersomnia. On the other hand, of course, classical sleep-deprived mice, they did not sleep, so they are sleepier. Okay, so we thought that we may cross-compare these conditions and take a common denominator, and that will indicate some biochemical change related to sleepiness. So turns out that sleepy mutation is in a serine threonine kinase called SIK3. We did a phosphoproteome analysis of the whole brain, and we found that a certain actually synaptically localized protein, the cumulative level of phosphorylation gradually increased with the increased sleep need, and recovery sleep will decrease it. So this is our current kind of model or hypothesis that a certain subset of synaptic protein, the cumulative phosphorylation, may represent some aspects of ongoing level of sleep need. So where is this sleepy mutation work? So this is obviously genomic, germline mutation. It turns out it's very interesting. If we recreate sleepy mutation allele in all the glutamatergic neurons in the brain, the sleep time increase and the sleep depth increase. In the cortex alone, sleep time does not change, sleep depth increase. On the other hand, in the hypothalamus, sleep time increase, sleep depth does not. Very interesting. And in addition, in the suprachiasmatic nucleus, the master clock location in our brain, actually the same molecular pathway regulates the timing of sleep now. So they could not get up in the early evening, which is equivalent to early morning in the humans. So another obvious question is, what is the molecular pathway involving this particular serine threonine kinase in terms of upstream? Since the SIK3 belongs to AMPK family, subfamily of kinase, in the literature it's well known that another kinase called LKB1 could be the upstream regulator. And indeed, this particular phosphorylation target for LKB1, this level of phosphorylation increases when the level of sleep need is acutely increasing. So this must be important. And indeed, the LKB1 deficiency causes the decrease in sleep time and decrease in sleep depth, or the level of sleep need. How about downstream? We continued forward genetic screen, and in the end, to make a long story short, we found that this loss of function mutation in HDAC45 could also increase sleep. And indeed, HDAC45 is actually a known target for SIK3. And indeed, this phosphorylation of HDAC45 by SIK is known to kick them out of nucleus so that they cannot anymore work as a transcriptional repressor. And indeed, the nuclear level of HDAC4, for example, decreases whenever mice are supposedly more sleepier, like after sleep deprivation, and in terms of daytime as compared with nighttime for nocturnal mice, and of course, in addition, the sleepy mutants. So currently, our current hypothesis or model is this LKB1 SIK3 HDAC45 kind of pathway may represent and regulate sleep, may represent the level of sleep pressure and accordingly regulate sleep. Amazingly, exactly the same pathway works to regulate sleep-like behavior even in C. elegans, we also found. So that's it for my part. This is just a one-page short acknowledgement, and I will switch gears to Sara's talk. Go ahead.
Thank you. I want to thank Dr. YANAGISAWA and everyone in the Moonshot Project who helped to bring me here today. As was true for Dr. Devlin, this is my first time in Tokyo and my first time in Japan, and I've been completely bowled over by what an amazing place this is, and I've been overwhelmed by the amazing science that I've seen here today. So what I'm going to be talking to you about is a question that's been long-standing in the field of sleep research, which is the relationship of sleep and sleep loss to neural functioning, and specifically to the process of storing new information in the brain. Now, this, as I said, is not a new question. It's one that has been studied for several years. I often start my talks by showing a picture of that was originally published about a century ago in 1924 in an early paper where the authors were investigating how well individuals in their lab could recall this list of just really nonsense verbal information that they encoded at time point 0. And then they waited different intervals of time and asked them to repeat back everything that they had just crammed into their brain. So think about this as like a true cram session and recall. And what they found in that study was that the amount of material that the subjects could recall and bring back into memory was a function of whether they had been allowed to sleep in that interval of time. So subjects that were allowed to sleep over varying intervals of time could repeat back far more of that information than subjects that were kept awake in the lab. Now, that type of study has been carried out many different times where human subjects have been given some sort of memory encoding task, some sort of learning task, and then they've been asked to perform that task later on after a period of sleep versus sleep deprivation. And so we know that this is a very common phenomenon, and it's also a common phenomenon in animal models. And I think very relevant to this audience, one thing that bears repeating is that as we get older, our ability to perform well on those tasks tends to go down, and also the amount of time we spend able to sleep and the number of nights that we spend with quality sleep goes down. And speaking from my own observations of myself, now that I'm becoming an older person, I certainly know that that is the case. It's worth mentioning, related to that as one of the leading predictors of cognitive decline in aging is actually individuals' habitual sleep time in middle age. So for individuals who habitually sleep less in middle age, their likelihood to go into cognitive decline in subsequent decades is much higher. Now, again, the relationship between those things is really unknown. It's not clear what the sort of chain of causality is. And one thing that my lab is interested in is basically what is going on in the brain during sleep, what are the underlying neurobiological mechanisms to help us understand why we need to sleep in order to store information in the brain, and why sleep deprivation is disruptive to that process. Excuse me. Now, there are a number of hypotheses in the field with regard to why sleep might be playing an important role in storing new information in the brain. A very prevalent hypothesis in the field, one that's received a lot of attention, is that over the course of sleep, if you look across the brain, what you see is evidence of general synaptic weakening. So decreases in synaptic strength, so weakening of connections between neurons. And the hypothesis here is that that process of general downscaling could be playing an important role for memory storage and retrieval in the brain simply by reducing signal to noise, if you will, between the signal of newly learned information and the connections that are being strengthened there, and then everything else in the background. Another hypothesis is that there is information content that is effectively being reinforced in the brain over the course of sleep through primarily reactivation of the same set of neurons that is active during learning and prior wakefulness. And related to that hypothesis is that the oscillations that are present in the brain in thalamocortical circuits during non-REM sleep and in hippocampal circuits during REM sleep, for example, are playing an important role in spike timing. And so that's actually reinforcing this process of reactivation of learning activated neurons. Forgive me, I'm trying to get my pointer out, but I'm hoping you can see that nonetheless. No, thank you. It's okay. Yeah, it's all right. I don't know why it's not working, but I'll just sort of like generally wave in the direction. So yet another hypothesis is that across periods of sleep, there is a rebalancing of excitatory versus inhibitory synaptic strength. And so what this simply means is that synapses that are corresponding to inhibitory transmission and excitatory transmission are undergoing differential changes, and that helps to sort of rebalance the network so you don't have overexcitation of the circuit during wakefulness. In addition, there are physiological changes. So one physiological change that's present in the brain, and probably many of you in this room are familiar with, is that during non-REM sleep in particular, there seems to be additional clearance of metabolic wastes from the brain via the vasculature, and presumably also delivery of nutrients into the brain. And so this could also be playing a role not just over the long term in processes like amyloid deposition, which it almost certainly is, but also playing a role in permitting the plastic changes that are associated with memory storage in the brain. And then finally, there are a number of cell biological changes that are happening in neurons and glia in the brain across periods of sleep versus sleep deprivation. And the function of these changes with regard to storing new information in the brain is generally unknown, but there are many of them that could either be permissive, that would allow for plasticity to occur preferentially during sleep, or that could somehow play some instructive role. Now, I'm going to talk to you about some of these aspects today. And the first one that I want to talk to you about is reactivation of memory traces in the brain. Now, all of our work is done in mice, and what I'm going to tell you about here is the work of Brittany Clausen, who's a very talented graduate student who graduated from my lab in 2020. Brittany developed a behavioral paradigm in which mice are presented with a visual stimulus. In this case, it's basically a grating of black and white stripes that surrounds them 360 degrees of a specific orientation. In this case, it's 45 degrees relative to vertical. She repeatedly pairs that visual stimulus with a mild foot shock through the grid floor of the chamber that the mice are in. And then she leaves them alone for the next few hours and allows them to sleep, or sleep deprives them over the first few hours following that pairing. And then at the end of the day, she places them back into the same arena and looks at their responses to either that same stimulus that was paired with the foot shock previously, or a similar but distinct visual stimulus. And what she's looking for is the animal's ability to discriminate between the stimulus that was previously paired with foot shock and this neutral stimulus. And what she found is that in animals that are allowed to sleep freely, they respond more strongly to the stimulus that was previously paired with foot shock than the neutral stimulus, indicating an ability to discriminate. However, when the mice are sleep deprived, they really perform at chance levels on this task. So they respond similarly to both of the visual stimuli. Now, to get a handle on where the information content in the brain is with respect to this and what role sleep is playing in reinforcing that information, we used a tool that was developed in Li-Chun Lo's lab at Stanford called Targeted Recombination in Activated Populations, or TRAP. So in the system, an immediate early gene promoter, so a promoter that is turned on by neuronal activity, is upstream driving a Cre recombinase that is fused to an estrogen receptor. Now, this will be turned on whenever the neuron is active. The mice in the study that I'm going to show you in just a second are expressing a second transgene, which is Cre recombinase dependent. Now, in the absence of neuronal activity, you don't have recombination because the Cre is expressed, but it's retained outside of the nucleus, so it has no effect. But when you administer tamoxifen or another agonist to bring Cre recombinase into the nucleus, you can get recombination. And so in this case, what you get is labeling of neurons that are activated in a particular learning context with TD tomato. So Brittany used these mice and the same type of paradigm where she paired a visual stimulus with foot shock. And in association with that, she looked at the effect of labeling the cells that were selective for that visual stimulus in visual cortex to assess whether those cells were being reactivated during post-learning sleep. So she has labeling with TD tomato, and then what she's done is either presented a foot shock for that same visual stimulus orientation or a distinct one. So this is basically controlling for the other effects, like, for example, the stressor of being repeatedly shocked. She waited a few hours, let the animals sleep freely, and then harvested brain tissue and just looked at the expression of immediate early genes in these orientation encoding cells in visual cortex. And what she found is that there's selective reactivation of that same neuron population when the prior learning is essentially identical to the stimulus that was used for the initial labeling. So what this is suggesting is that in this offline period after the learning, you're getting reinforcement of the information that was just encoded hours before, and in this case, the pairing with the foot shock. Now, to see whether this was actually necessary for learning on the task, what she did instead was expressed archirhodopsin, so an inhibitory light-activated protein, to suppress the activity of these cells. And what she did was deliver light into visual cortex during post-learning sleep periods in animals that had had this recombination to the 45-degree stimulus and then were either shocked, paired with the 45-degree stimulus or a different stimulus. Then she looked at the memory consolidation in these mice, the ability to discriminate between the two different stimuli. And compared to controls, what she found is that in mice that had undergone this light-driven inhibition of the memory encoding population, there was a disruption of memory storage. And in mice that had undergone a completely different learning pairing, there was no effect of that same inhibition. So this suggests that what's going on during sleep is that there's some selective reactivation, reinforcement of the information that was just encoded. Now, we got really interested in what this process might mean for structures like the hippocampus that tend to undergo neurodegeneration with aging and that are extremely important for the types of memory that tend to get selectively degraded with poor cognitive aging. And the reason that we were interested in the role of sleep in particular on this is that there was beautiful brain imaging data from human subjects doing hippocampus-dependent memory tasks where the effect of prior sleep deprivation was assessed on brain activation during the task itself. And what was found is that in animals that are sleep deprived, in subjects that are sleep deprived, they are animals. What was found is that the overall activation of that structure was suppressed, meaning that the hippocampus simply couldn't activate and engage in the same way during performance of the task. And furthermore, if we look across studies using human subjects where effects of sleep deprivation on memory have been assessed, across a wide range of hippocampus-dependent tasks, spatial tasks, associative tasks, you see a deficit after a period of very brief sleep deprivation after the learning. Similarly, with rodents, you see the same sort of degradation. We were interested in using a pretty well-studied model of memory consolidation, which is contextual fear memory. Now, in this task, mice are placed, rather than having a pairing with a visual stimulus, they're placed in a novel context where they receive a foot shock. If you then replace them in that same environmental context, it can be a day later, it can be a month later, what you find is that there is a pretty standard response that the mice will exhibit, which is freezing behavior, very similar to what we were measuring with the other task. Now, if you look at that freezing behavior at recall in animals that are sleep deprived, we and others have found that if you deprive mice of sleep just in the first few hours following the single context foot shock pairing, you see a major decrease in the animal's response to that same environmental context, meaning that there's less of a fearful association with that context. Now, interestingly, in the same few hours window of time where animals need to be sleeping, you also need to have the hippocampus being active. You have to have activation of a series of kinases. You have to have de novo transcription and translation. And so a question that we were interested in addressing is, how is sleep mediating activity-regulated biosynthesis in the hippocampus? And to get at this, another graduate student in the lab, James Delorme, became interested in phosphorylation of ribosomal subunit S6. So S6 is phosphorylated by a number of kinases in neurons that are regulated by the activity of the neurons. And it's thought that phosphorylation on a series of serine residues on the S6 subunit is not necessarily changing the overall rate of translation at the ribosome, but is changing the specific transcripts that are being translated. Now, what James found right away, which was rather dramatic, is that if you sleep-deprived mice for just a few hours, you see a huge reduction in the overall level of S6 phosphorylation across the hippocampus. So in as little as 3 hours of sleep deprivation across the 3 major subregions of the hippocampus, you see a reduction in phosphorylation. And to get a better sense of what this means with regard to transcripts associated with those ribosomes, and also with the overall activity level of the network, we used this, the antibody we were using here for immunohistochemistry, as an affinity tag to pull down ribosomes and associated RNAs. We then looked at transcript clusters that have expression correlated with the amount of prior sleep time. And we found 2 clusters of co-regulated transcripts whose expression is decreased in proportion to the amount of sleep time. So more time spent awake, more sleep deprivation, more expression. We then did cell type specific expression analysis to look at cell types that might be represented in this phospho-S positive population of neurons that remains after sleep deprivation when overall levels of S6 phosphorylation are going down. And what we found was a couple of hits that were not surprising at all. First, we found that cholinergic and hypocretinergic neuron markers are expressed at higher levels when animals are sleep deprived. The reason this isn't surprising is that both of these cell populations send dense projections to hippocampus, and both cell populations are known to be wake active. So these are ones that Dr. YANAGISAWA just mentioned. However, we also saw markers of interneurons popping up, and specifically interneurons that are expressing neuropeptide Y and somatostatin. Now, these cells are interesting because they send strong inhibition to the principal cells in the network, so the excitatory neurons, and also other interneurons. So they provide really strong inhibition to their neighbors. And we used immunohistochemistry just to verify that we're getting increased expression after sleep deprivation in structures like the hilus of the dentate gyrus, where there's a very large population of these cells. And that did seem to be the case. And because these cells are providing really strong inhibition to their neighbors, we were curious as to whether this activation during sleep deprivation was actually suppressing the activity of the surrounding neurons in the network that would basically be providing the output of the hippocampus. And so to mimic the sleep deprivation condition, we first expressed HM3DQ, which is a DREAD for chemogenetic activation of the somatostatin neuron population. And what we found is that in animals that are expressing HM3DQ, when you activate this cell population immediately following learning, you wind up getting very strong activation of those somatostatin-expressing interneurons in the hilus. You also wind up getting suppression of activity markers in the surrounding excitatory neurons, principal neurons. Moreover, if you look at what this does to consolidation of memory after training, you see that it is disruptive. Now, we were also curious if the converse was true. So if you inhibit the cell population, do you get better memory consolidation? And in fact, that is the case. Forgive me for rushing through this for the sake of time. So this suggests to us that this cell population is providing an inhibitory gate on the surrounding excitatory neuron population in the hippocampus. Now, just to follow up briefly on other findings that we had from this larger study looking at transcripts associated with phosphorylated S6 ribosomes and also ribosomes from pyramidal neurons, the excitatory neurons in the hippocampus. This larger study was looking at transcripts associated both with ribosomes in chem kinase expressing excitatory neurons, using ribotag, which is basically a Cree-driven affinity tag. And in the same animals, we were using that antibody to profile phospho S6 ribosomes. And then also we looked at the overall hippocampus. Now, in the study, we had a 2-by-2 design. One factor was the learning condition. So some animals stayed in their home cage, some animals underwent fear conditioning, and then sleep condition was the second factor. So some animals in each group were allowed to sleep, some were sleep deprived. And we did sequencing on everything and just looked at some of the interactions between learning and subsequent sleep. And for the sake of time, I'm just going to very quickly go over what was months and months of analysis by my student, just to show you the networks of transcripts that are affected following learning, depending on whether an animal is allowed to sleep or not. So first, I'm going to talk about the excitatory neurons in the network, the ones that are being shut down by sleep deprivation. So if following learning animals are allowed to sleep, you uniquely get upregulation of pathways that are encoding components of extracellular matrix. Why is this significant? Because we know extracellular matrix remodeling is an essential part of synaptic plasticity. You don't see this in the sleep deprivation condition. However, you do see regulation of a number of different cellular stress pathways. So unfolded protein response pathway, ER stress, autophagosome maturation, all of these things are being affected specifically and selectively in the sleep deprivation condition. Also cellular energetics, so transcripts that are encoding components that are essential for providing energy to the cell are all being downregulated selectively in the sleep deprivation condition, not in animals that are allowed to sleep. In the phosphorylated S6 population, what we see is something very different. The first thing we see that we think is very interesting, specifically when animals are allowed to sleep, but not when they're not, is that you have upregulation of transcripts that are encoding the protein synthetic machinery. So a number of ribosome components, et cetera, are being upregulated only after learning that is followed by sleep, so only when consolidation is happening. Now, on the other hand, in the sleep deprivation condition, what you have is upregulation of a number of components that are required for increasing synaptic inhibition. So consistent with the other data that I showed you before, it looks like synaptic inhibition is causing some sort of gating on consolidation. This is just a different way to look at that same data. Also, neuroinflammatory pathway components are upregulated, specifically in the sleep deprivation condition and not in the freely sleeping condition. So just to summarize what I've just talked about today, so what we find is that sleep-associated reactivation of specific neurons in the brain immediately following learning is an essential component of memory storage. Sleep disruption seems to inhibit this process by gating the activity of those neurons through inhibition, by essentially increasing the activity of inhibitory neurons, which in turn suppress the activity of the information encoding cells. And then finally, if you just take a zoomed out approach of transcripts that are changing, what seems to be happening in animals that are allowed to sleep after learning is that biogenesis and extracellular matrix remodeling are potentially enhanced. On the other hand, sleep disruption seems to disrupt really essential cellular processes, such as cell energetics, and seems to activate neuroinflammatory signaling. And again, this is with very, very brief sleep deprivation. So with that, I just want to thank the people who contributed all of the data, James Delorme and Brittany Clausen, and the rest of my team who provides support for our lab every day and keeps us coming in and keeps us smiling when we leave work every day, which is very important. And I'm happy to answer any questions you have.
Thank you for the presentation. We will now proceed to the Q&A session.
Okay, so to start with, I have a kind of very generic question, especially for this audience. So it's not directly related to your own work, but in one of the introductory slides, you showed us that in humans also, sleep is already shown to be very important for various modalities of memory. I remember nearly 20 years ago or so, Jan Bohn's group described in a Nature paper that what they call an insight is also enhanced by sleep. I mean, this story is so interesting. So the individuals are given for this little quiz with numbers. And this quiz could be sequentially solved by step, but it takes a long time. But once you notice a hidden cue, hidden to the problem by itself, you can immediately solve. And they found that after a number of training before sleep, the slept group, this what they call insight, this shortcut, the rate of those individuals who can find the shortcut significantly increase. With sleep deprivation, it never happens. So, sorry, my question maybe you may or may not know, but that type of, kind of very high level skill or memory, not even a memory, right? High level skill is enhanced by sleep. Is that holding today?
I think that's one really great example of, essentially it's a type of implicit learning. And I would argue that a lot of motor tasks probably rely on that same sort of thing, where there's a trick to it. And it's not stated. You can't easily tell someone how to do it, right? But eventually something clicks. And I think for the sensory motor tasks, that's very clear. Like there are a lot of studies using all kinds of different paradigms where they're finding effects of post-training sleep on the ability to, at the very least, perform tasks that rely on some sort of implicit adaptation, rule learning. And my sense is that, and again, I'm speculating wildly here because I think the neurobiological underpinnings of that sort of learning, I think we have a long way to go still to understand that. But what I would say is, in all of those tasks, what you're going from is like making a very simple association of, 1 + 1 = 2, to sort of A + B = C, then A = C. And I think to do that, you just have to engage more. I think you just need a larger network. And so, my sense is like what sleep might be really good for is those sorts of things where you're sort of building things out. I think another like analog of that is like schema learning. So this is from like the psychology field, but like schema learning or any sort of like implicit rule learning. There's been evidence where people have done studies like that and found benefits of sleep. Now there's argument about like which stage of sleep. Is it non-REM only? Is it REM only? Do you have to have both? And I think our understanding of the roles those two states play are going to evolve. It's going to evolve like as we know more about what those states are doing for networks in the brain.
Yeah, another obvious question, your data shows that some of the transcriptomic change involves this genes involved for extracellular matrix synthesis. That's amazing. And I seem to remember recently I either saw a paper or heard a seminar by Paul Frankland lab, who also describes some changes in extracellular matrix. Is that correct? Related to memory? You know.
I don't know if
anyway.
was it published. I hope.
Maybe it's not published yet.
I hope so, because it's I think it's very affirming when another group using a different set of paradigms.
Anyway, so do you have any speculation of what's going on there?
Yeah. Okay. So it's a really great question. I think I would say a couple of things. So for the data I showed you. Okay, let me back up. So I think what we're seeing here, and possibly what Paul Franken's lab is seeing, might be related specifically to the interface of learning and sleep. Because we definitely see things emerging there, and that's specifically where we see that set of transcripts being upregulated, where there's a definite need. I guess I don't know. It seems like anthropomorphizing, but there's a need of the network to have those molecules be generated in order to complete the process of plasticity that gets initiated during prior wakefulness. And whether that is a general feature of sleep, I'm a little bit more skeptical. So I think for a lot of these studies, either the effect of learning is being looked at in isolation or the effect of sleep in isolation, and it's hard to make assumptions about the interactions purely based on those.
Okay.